Welcome everyone to the April meeting of the Science Engineering Council. We're here the second Wednesday of every month with our speakers from around the community and the science engineering practices. So looking forward to today's discussion. Before we do that, just acknowledge our sponsors. You can see them on the screen there: Community West Bank, Minnow Enterprises, Bengal Engineering, and Toyon. Feel free to reach out to any of the board members here or send us a note if you'd like to sponsor or become a corporate member or of course, as an individual member. That's vitally important. Board of directors here, I've got myself, Tim Murphy, we got Yvonne DeGraw, our program chair. Also on today's meeting, we have, Mary Howe Grant, John Fishpaw, I see Joseph Stronks, Bob Lilly, Bill Theringer, and our corresponding secretary, Cheryl Ebner. So with that, just want to thank everyone for the participation in our special meeting we had a couple of weeks back on the Science Fair Awards. That went very well. A little tricky doing it online, but we did it. And those prizes went out to the worthy winners of those Science Fair awardees. Okay, so with that, I'll turn it over here to Yvonne to talk about upcoming meetings, and then we can kick off with today's discussion.
Great, thanks, Tim. Hi, everyone. We have a full slate of upcoming talks scheduled through August, and hopefully we'll return to in-person luncheons sometime soon. Our next meeting will be on May 11th, and Dr. Kenneth Kozik, co-director of UCSB's Neuroscience Research Institute, will speak on the surprising science of brain organoids. On June 8th, Dr. Rachel Street from Las Cumbres Observatory will speak on hunting isolated black holes with microlensing and some exciting black hole discoveries that they have contributed to recently. On July 13th, Dr. Galen Moody from UCSB's Electrical and Computer Engineering Department will speak on quantum computing and quantum photonics. And on August 10th, Dr. Christopher Richardson from UCSB Molecular, Cellular, and Developmental Biology will be speaking on DNA damage repair. And today, we're happy to have Dr. Michael Mahan as our speaker. Professor Mahan is in UCSB's Department of Molecular, Cellular, and Developmental Biology also. He received his PhD in genetics from the University of Utah and was an NIH postdoctoral fellow at Harvard Medical School, where he began his work on the molecular mechanisms underlying salmonella pathogenesis. Dr. Mahan joined the UCSB faculty in 1993 and was a co-founder and director of Remedyne Corporation, a biotech company in Santa Barbara. You can post your questions in the Zoom chat during this talk or ask them during or after Dr. Mahan's talk as appropriate. So Dr. Mahan, go ahead. I'll stop sharing here.
Well, first I'd like to say thank you for having me. It's always exciting to meet new people, and obviously you guys have done your share in the scientific world, and so it's actually I have three children, two are in engineering. One's getting her PhD in mechanical engineering and robotics at Rice University, and we have a son that's a sophomore at UC Davis in mechanical engineering as well. I also have another son who's getting his PhD in infectious diseases, kind of similar stuff that we do. But anyway, well, thank you. And I'm going to tell you a little bit about a new diagnostic we've developed. It's a smartphone-based diagnostic, and it's among the most rapid, sensitive, affordable, and scalable tests known. And it can be done in a fraction of the time and cost of current PCR testing. And it also provides a platform for home-based testing. And we're going to talk a little bit about that. And I'll give you just a general description what's going on. Basically, we have an app and a lab kit that we've developed. And basically, the smartphone's camera takes a picture of a chemical reaction in a test tube, and it determines the diagnosis in 25 minutes. The chemical reaction contains a fluorescent dye. And basically the bottom line, and as you amplify, you get more glowing, and more glowing, more virus. And the test costs less than $7 versus for each test versus $100 to $150 per PCR test. And we think we actually can to drop that cost significantly. We've provided all of this information, the technology, the app, the code, everything free, open source, freely available to all, just trying to do something good for people that are struggling with the pandemic, which there's many. So. Advance this. Here's some COVID facts as of yesterday. There's been 498 million cases globally, 6.2 million deaths. Currently now 58% of the world is vaccinated. Vaccinated being defined as having the two initial shots, not the booster. The United States, we've had 80 million cases, or almost nearing 1 million deaths. Embarrassingly, actually, for me anyway, that almost 1 in 6 deaths are in this country, where we have some of the best, if not the best, medical care in the world. We have 64% that are vaccinated, and which is also somewhat hard to understand for me. The science is in. There's been 11 billion doses given of the vaccine, and it's safe. I think this is the biggest human experiment ever done. Oh, I'm having trouble advancing here. Let's just try this. Okay, let's talk about what's out there now. The gold standard by far is PCR. Okay, it's super sensitive, it's super accurate, but it's very slow and it's very expensive. It's $100 to $150 a test. This will never be a home-based test. PCR has been around for a really long time. If it could be a home-based test, it would be a home-based test, and it's not. We're going to tell you about a technology that's not new. It was developed in 2000. It's called LAMP, which has a long name. It's called loop mediated isothermal amplification. And what the key part is, it's isothermal. What that means is that you have the whole reaction occurs at one temperature. And that's something that's really important if you want to have something that's inexpensive and widely available. PCR, one of the reasons why it's so expensive is that you need a very fancy machine, and it goes to this thing called cycling, where it goes to very hot temperatures, very cold temperatures. You have to separate the DNA strands. You have to have a very sensitive instrument to be able to do that, and you have to have you have to pay someone who's highly trained to be able to run it. LAMP is not that way. It's an isothermal amplification. Basically, you add reagents and you go away and you come back and you get your answer. Now, this has been around, so I guess, again, as I said, since the year 2000. And why isn't it used now? The reason is it's ultra sensitive. It's 10 to 100 times more sensitive than PCR. Because it's so sensitive, you actually get it's inaccurate results, because you'll get these false positives where the components of the reaction start amplifying themselves. If there's no substrate, there's no virus around, it'll hunt for components, and then it'll amplify. And this has been called the primer dimer problem and we'll talk a lot about that, but it's been a problem in science for 22 years, actually. And last is the rapid antigen test. It's fast and low cost, which is great, 10 to 12 bucks, but it's very insensitive and it's very inaccurate. Okay, we have a lot of false negatives. And for me, it's a little frustrating in that I think they're being massively, widely misused, and that they have a function. And what the function is if you're a rapid antigen test positive, you are positive, okay, period. Okay, but if you're negative, because it's so insensitive, you don't know if you're negative, okay? And so what people do, in fact, it happened in my own family. My brother, his wife's, I don't know, 50th birthday was New Year's Eve, and they had a big extended family, and they all met in the garage, and they all had rapid antigen tests, and everybody took them, and everybody was negative. Well, the next day, someone had symptoms, and every single person at the party, everyone got COVID. I mean, it's, and I said, my brother's name is Pat. I said, Pat, why didn't you ask me? That's what we do for a living. And he goes, oh man, my bad. And so basically, but this is, and I'm not sure why that's not out there in the news, but it has a function. It's a function to see if you're positive with a lot of virus. Okay, but it's not great at, it's not in the league of PCR. It's just, they're just completely different. Okay. Okay, so I'm going to tell you about this new technology. It's called Smart Lamp. And basically, it's a smartphone-based lamp test. I'm going to tell you about how we solved the primer dimer problem. I'm going to tell you about its ultra sensitivity and specificity. I'm going to show you some evidence that we've now done a clinical evaluation with patients from Cottage Hospital. And where we show 100% concordance with the RT-PCR test. We've now, our initial vision was to have this for point of care for resource limited nations. And so we've spent a lot of time developing conditions where we can, RT stands for room temperature. This whole thing can happen at room temperature. You can store your specimen at room temperature. You can assemble the reaction at room temperature. And then you now do your actual reaction at a constant temperature. So you don't need freezers. You don't need things that are expensive. And I'm going to leave you with some applications to point of care testing, home-based testing, and also variants. Pandemic variants like the flu. Let me get something. This is a horrible pandemic. Clearly, this is nothing compared to the 1918 Spanish flu, where 60 to 100 million people died in 1 year, and there were no airplanes. Okay, and so this is not. I can tell you, I used to consult for Lawrence Livermore, which is the bellwether, the United States' bellwether for biodefense, and I went up there a lot, and believe me, I think I went out there right after 9/11, like the next day or that day, I forget, and we were trying to figure out what happened and what we're going to do, and basically the flu. This is a horrible pandemic. I'm not trivializing this, but if now with all the jet travel, et cetera, we need protection against things like the flu as well, in addition to coronavirus. And so we think we've made some inroads into that as well. Okay, so what is this primer dimer problem? Again, this technology has been around since 2000, and again, it's ultra sensitive. And so what that means is that to be able to amplify any kind of a nucleic acid like RNA or DNA, you need it's called a primer, something to start. They bind to the bases and it'll start amplifying. Those are called primers. And so the problem with this technology, it's so sensitive that if there's any homology between the primers themselves, they'll amplify that if there's no virus around or no bacteria around. It's that sensitive. And so we knew if this was going to work, we're going to have to solve this problem, and it's been around since 2000. And so what this is just old school biochemistry. There is nothing fancy about this. It's a matter of just sitting down, and just I'll tell you, it took a year and a half, 7 days a week, no days off, no birthdays, no Christmas, no New Year's, no nothing. You know, we were in a race and we wanted to get it done. And we had a team that was willing to do that. And so what it was, it's like, here's what we had to worry about. Here's just some of the things. There's probably at least 10. But the pH of the reaction, the temperature of the reaction, all the different salts you put into the reaction mix. What are the concentration of the enzymes that amplify? What are concentration of these primers that start the amplification? You know, you have to have RNase inhibitors because in saliva, for instance, there's it's naturally made to break down RNA and DNA. It's a protective response. You have to stop that. And also, will it work in saliva? Actually, we were one of the first to use saliva. I think that the nasal test is great, but it's uncomfortable. And we thought saliva would be the way to go. And so what the problem was like a giant Rubik's cube where you're manipulating all of these variables are connected. And what we found was not only the order, the composition, but it's the order in which you added them. That was also important to be able to solve this problem. And so basically it was like a giant Rubik's cube. And when you'd fix the yellow side, the green wouldn't be, add up. And then you'd fix the green, the blue wouldn't add up. It's like whack-a-mole, however, we were just determined and we just kept at it. And it took us 500 tries. Theoretically, there's was 10,000 different possibilities. We got it by just thinking about, narrowing it down basically from 10,000. We got it down to 500, which took us a year and a half. And when we got it, it was just different. And the next day I said, let's try it. Let's see if we can distinguish the flu from COVID and SARS-CoV-2 is the virus. And the very next day, on the first try, we diagnosed and distinguished 2 different strains of the flu using this technology. So then we knew, we had solved this problem. And so here's a picture of what we see. That basically the camera is taking a picture of this fluorescent reaction in this little, we're called microtiter plates. Every 10 seconds, the camera takes a picture, and we're now going reaction time. And what happens for a positive sample, you can see between here and here, doesn't matter what this is, but basically you can see you get amplification, and this is again by your phone. And if you have no virus, you do not get amplification. Now with the primer dimer problem, this thing would spike later, like about here, and it would confuse the results. So by getting the pH, the temperature, the salts, the enzymes, the primers, et cetera, correct, and not only was it finding out the concentrations, it's when and how you added them. And we provided this recipe, it's just like making brownies. We provided every single step in our paper, free, open, for open source to everyone. And you can see we don't get any primer dimers anymore. So we've eliminated this problem that's been around for 20 years. And so what this means is now this very cheap, this very fast technology can now be used. And it's not only for a smartphone, but for any instrument. Okay, so this LAMP technology now, you can use it in a PCR machine. You can use it, whatever machine you have. Okay, so I'm going to tell you, here's the features of the smart lamp. The diagnosis, we can make a diagnosis in 25 minutes. Actually, a positive can be detected in 15 minutes. It's why, as opposed to PCR, it'll be hours, 2 days before you get your results. It's ultra sensitive. 1,000 copies per mil is the barrier of some of the most sensitive amplification tests in the world. It's very affordable. It's less than $7 a test. And I want to be clear on this. This is $7, less than $7. Actually, it's $6.47 a test at full price. Okay, we didn't get any deals on this. We bought them at small quantities. There's a lot of room to be able to drop the cost of this test. And it's importantly for resource limited nations. It's highly scalable. You can test massive populations in that 96 tests per run per phone. So just think about that. It's 25 minutes. Every 30 minutes you turn it over. You can have a bank of 10 phones. You can do the math. I mean, you cannot do this kind of scalability with PCR. You can't afford it. I mean, a PCR machine, each machine is $35,000 to $60,000 each. Okay, we got these phones off Amazon for 150 bucks. Okay, so we have a little lab in my little test lab in my where we can run and do these scalable tests. This was published at the end of January this year in the Journal of American Medical Association, their open journal. And let's get to it. So what you need is very, very simple. You need some LED Christmas lights, some LED lights to illuminate the fluorescent dye. You need a cardboard box, which is here. And you need a heat block from Walmart. That's what you need for this. You can fabricate this for less than 100 bucks. Actually, the cost is actually $80 of the 100 was just the cost of the sample holder to hold to have something that would transmit heat from the test tubes to the hot plate. That was $80 of the 100. So we think we can get this down to 20 bucks. We imagine in the future, if we do home pain testing, this is something that you would buy at CVS or whatever, Longs or wherever you are in the country. And so again, and then your smartphone is basically put on the top. This is a cardboard box. You just basically put a hole in the top of the box and you just put your phone down and you turn it on and you walk away. Okay, so what's the assay workflow? Basically, you spit into a tube. Okay, you add it to this, we call it our magic mix, this reaction mix that, and that's actually one of the key findings is what's in the mix and how is it added. Okay, so that's there's no magic. It's just the like there's good cooks and bad cooks, right? Good brownies and bad brownies. Well, this leads to the good brownies. And as you get and this fluorescent dye glows when there's virus, and the more glowing, the more virus. And so on your phone, this is what you see. Okay, again, really, let's review this. Okay, you spit into a cup. You pour the spit into a reaction mix. You put it on a hot plate. You turn on your phone and you leave. You come back 25 minutes later, and this is what you see. If you don't have any COVID, you're green, and so we have a little check. You're good. You're checked. Good, good, good. And here, if you find a red button, you're not good. You got COVID. And not only can we detect at one of the highest sensitivities known of any assay known, not only can we detect, we can quantitate. We can tell you how much virus you have. So if you, and I'll show you on the next slide, but if you click on a red button, your detailed results will come up and it'll tell you how much virus per mil do you have. And again, this is done in real time. And so basically what we imagine at resource limited settings, it's important to be quick, super cheap. You can't say, oh, you come back tomorrow, and get your result. This is not happening. It ain't gonna happen at home-based testing either. You wanna do it just like a rapid antigen test. Everybody does them because they're cheap and they're easy, right? So you can detect and you can quantitate. Okay, and I won't go into the super details of this, but basically this is what you see. This is our app that we've now provided freely available online. Basically, we've done this technology we've published before. It works in blood, urine, feces, and then saliva for COVID. All you gotta do is click what bodily fluid you have, and you hit record, and you go. There's nothing else. Okay, I'm not a coder. I'm not a computer scientist. I'm not an engineer. Biologists. It's dumbed down so much that even mice, I can understand it. Biologists we like things simple. Okay, and so basically you click the record your sample and you leave. 25 minutes later, this will pop up on your screen. Again, if you're negative, you're going to be a nice check mark. These are each individual sample here. These are different people. okay, different patients. And then, however, if you're infected, then you have a red button and you press on the red button and it'll tell you exactly how much virus you have. Well, why is that important? Well, you could be a super spreader. You could be on your way to recovery. You want to know, physicians want to know where you are in this cycle, okay? Because depending, basically on for transmission purposes, okay? So we have that covered. Okay, so now I'm going to tell you, I'm going to go through this slowly. We're going to go through the sensitivity, how sensitive this test is, and we're going to go about specificity, which means if you're looking for COVID, you get COVID. Okay, if anything looks like COVID, well, you get that too. That would be called a false positive, and you just can't have that. Can't have a false negative. Look with the rapid antigen tests, false negatives all the time. Everybody at the party gets sick. You don't want false positives either, because people get freaked out because they're not sick and they think they're sick. And I mean, that's the thing about you have to be sensitive and accurate. You got to do both. Okay, so let's look at about sensitivity. So here we have smart lamp, and PCR is really the formal name for it. It's RT-qPCR. This is this say PCR. That's what this is. Okay, that's the green boxes. And here we had 1,000 copies of virus per mil, which is in the elite category for sensitivity. There's lots of RT-PCR tests, just like there's lots of brownie recipes. I'm talking about this is the best PCR test. There's lots of PCR tests that are less sensitive. This is the best. This is what the CDC gives or you buy. And at 1,000 copies per mil, this is the smart lamp versus the PCR. At 500 copies per mil, this is smart lamp versus PCR. So it's highly sensitive. This is called the limit of detection, LOD, the limit of detection for SARS-CoV-2. Okay, that's great. So it's very, very sensitive. But how specific is it, right? You don't want false positives, okay? So this paper was submitted in December 3rd, 2021. So at the time, Omicron, when we submitted the paper, wasn't around, okay? And so what we tested was, we'll go through this carefully. Now this is specificity, this is important, okay? Will this test only amplify SARS-CoV-2, if that's what you want to, if that's what the primers are targeting? The answer is yes. And so we're going to go through the evidence. This is SARS-CoV-1. I don't know if you remember the first SARS scare was in 2003. This came out of China. And high mortality rate, et cetera, but didn't get transmitted like CoV-2. It was, things got shut down and they contained it. But that was in 2003. 2012, another similar coronavirus called MERS, Middle Eastern Respiratory Syndrome. That came down in 2012 in Saudi Arabia. This is, it jumped from camel to human. Well, there's no detection of these similar coronaviruses. Now, when you look at SARS-CoV-2, this is a USA strain, a Hong Kong strain, the Alpha variant, the Gamma variant, the Delta variant, the Epsilon variant, the Iota variant, detects all of them. And I can tell you, by Omicron wasn't around, it didn't exist. You couldn't get samples in the beginning of December, but we can do what's called bioinformatics. We know what targets our primers amplify, and I can tell you that that this test, as it is now with the current primers, the current test kit can detect every variant known. Okay, and if something happens, I'm a geneticist. This is my formal training. We thought hard about what target we're going to amplify. We stayed away from the spike. We knew that sucker spike was going to mutate. Okay, because that's what binds to the spike protein, is what binds to the receptor. We knew that the virus was going to mutate, and so we chose wisely, basically. And still, we haven't found a variant we can't find with the current technology. And the thing is, let's say one happens, and it will happen. You just change the primers. Everything else is the same. All the conditions are the same. Okay. Here's four. We get coronaviruses every year. These are called cold coronaviruses. These four human, that's what it stands for, human coronavirus. They're called it's common cold. 1, 2, 3, 4. It does not recognize any of these. What about other respiratory viruses that when you spit, what if you have the flu? Will you get that? No. What if and if you have another, there's two main classes of the flu A and flu B. No. What about strep pneumonia? What if you have a pneumonia infection? No. What about if you have a staph infection? No. Pseudomonas infection? No. Klebsiella infection? No. So this is highly specific for only SARS-CoV-2 and its variants. Okay. Well, as I said, I alluded to at the very beginning, someone I consulted many years for counter-bioterrorism for the United States, and I can tell you what I'm worried about is the flu, the influenza, because that tests 60 to 100 million a year. 1918, that flu killed more people than the entire. This is at the end of World War I. It killed more people than both sides of the war in less than a year. And the reason for it is that the flu is it's a nasty virus. It has 8 chromosomes. It's like human. We have 23 chromosomes. This virus has 8, and they're constantly mutating. And what we're worried about is that there's a set of transmission genes on, let's say, one chromosome, and the virulence genes are on another chromosome. What we're worried about is that we get mutants that are highly transmissible all the time. We also get mutants that are highly virulent all the time. And it's just like I can tell you, they're basically every year in China, or at least every other year, there's a new virulent variant of influenza that'll kill 50% of the people it infects. Think about that, the flu. Okay, can you imagine the flu if it's transmissible as the current common flu, and now kills 50% of people that it infects? So what they do, they kill every duck, every goose, every waterfowl. They kill millions. They kill all of them, and they start over again. That's what they'll do in 2022. The reason for it is that the flu is carried in waterfowl, migratory birds, and they don't get the flu, but they carry it in their intestines. And what they do, they poop on. It's carried in their poop, and then and this is how it's transmitted. And so how they deal with it, they just kill them all when there's an outbreak. But people have died in these small villages, right? Half the people in the village die, and they go, okay, we have to kill, every duck, every goose, and within whatever so many miles of the place. And and currently that's what's done. Actually, I went over in 2014, I went over to the Republic of Georgia, and I was supposed to speak on this problem. And what I said was, you got to stop these open markets where the ducks and the geese and the monkeys and civet cats and the humans are all together. Now, I'm not a sociologist, I'm a scientist. I realize that is a huge problem. Their parents, that's what they do. They sell live animals at a market. Their parents sell live animals at a market. Their grandparents sold animals at a live market. I get that. I'm not a sociologist. I don't know how to deal with that. But they asked, I wrote a thing about it and said, well, yeah, you have to stop that, because that's how these things are occurring. These free range chickens that are not, so basically anyway, in a situation where a duck or a goose can poop on your chicken, it's going to get infected, and it's potentially being infected with one of these super strains. Okay, so here's the flu. So now again, I'm going to tell you, we now change the primers to detect specific strain, either flu A or flu B. So now it's the same lab kit, identical lab kit. As I said earlier, we did this the very next day after working a year and a half, 7 days a week to get COVID for one time to work. The next day we got this the first time, and then the second time, and the third time, and the fourth time, with the same exact kit. And the only thing we switched out were the primers that basically we're now targeting different genes that are specific for influenza. And again, this is smart lamps, the blue, PCR is the green. And we can show that when you have flu A and you have flu A primers, you can detect it with smart lamp 100%. nearly 100% with RT-PCR. If you don't have the primers, you don't find anything. If you now, basically you can find flu, sorry, the blue is flu A, the green is flu B, this is smart lamp, and this is RT-PCR. Basically, just look at the big box, the big buildings, you detect it, okay? So what about the specificity for influenza? As the case for here, for COVID, if you now are smart and make the right primers that are specific for that, you know, for that, and there's actually programs, you don't have to be that smart, I mean, there's programs that'll do it. And you can say if you have influenza primers, blue, you find influenza A, but you don't find influenza B. If you have primers for influenza B, you detect it, but not influenza A. Okay, so this so it's highly sensitive for influenza and highly specific for influenza. You know, it doesn't detect any coronavirus, doesn't detect any strep pneumonia. This is stuff you'd find in your saliva, basically. That's why we tested this. So what we've shown, now this is all done in the laboratory now, which is the way things are done. You do things in the lab and then you do the best you can, and then you go into, you do the human stuff. Okay, so, and I'll take, there's a chat. Why don't we take a chat question? Does someone want to read it, or I guess I can click on it. Yes, is the fluorescent dye? Yes, a very good question. The question is the fluorescent dye the same for both COVID and influenza? Yes. Yeah, it's called calcine. It's just a cheap fluorescent dye. I'm sure there's better dyes, but it's super cheap, widely available. And that's the whole thing, is that the only difference between these two experiments is the primers, and they're just what starts the amplification reaction. So that was a really good question, actually. Okay, so now it comes to, let's see if we can really do this. And so our collaborators are our physicians at Cottage Hospital. We've had a long time collaboration with them over a number of projects. And actually it was, you know, they're very, very nice. They're super busy. We did this during the peak of COVID, where they, you know, they're in their full war gear, battle gear, and I'm in my office, you know, with my shorts and my t-shirt on. And, you know, they took their time. They'd go in basically a small room, and they would, you know, and so we said, hey, are you, you know, we think we have this test. Can you help us? And they said yes. And so what the story is that the question is lamp, is this smart lamp, this new technology that's we use in the smartphone, is it as sensitive as PCR in actual patient samples? Okay, so we didn't sample the patients, the physicians sampled the patients. Actually, we couldn't, then the hospital was locked down. We couldn't, I'd go over there, they wouldn't let you in. They would actually come out, the physicians would come out with a little thermos full of samples, and we would come back to my lab and we would check them. And so the bottom line, I'll tell you the answer first. The answer was we had 20 PCR positives. So PCR tested positives. This was done blind, so we didn't know the answer. We got 50 samples. And you know, once we now decoded and whatever, there was 20 positives by PCR. They're up here, 1 through 20, and they're labeled from patient 1 through 50, but 20 of them were positive by PCR. The same 20 were positive by our smart lamp. 30 of them were negative for the PCR, and 30, so here's the PCR, and the same 30 were negative for the lamp. And so what we wanted to do was to also test for influenza. And because of the social distancing and hygienic measures we've taken, i.e. masks, washing your hands, staying away from people, etc., there was not one single case, hospitalized case of the flu. Not one. Okay, so basically, so these 50, 1 through 50 here, patients, that basically none of them had the flu, influenza A or influenza B. They're all below the detection. Now, when we're now relaxing our at least at UCSB, we're relaxing our, I still wear a mask, I'm at home right now, but you know, this is what we study, and I can tell you this ain't over. Okay. I'm glad everybody's getting a little reprieve, but you know, at least I think the data is still out. But one thing that people don't know, and I think that I'm in the same. I'm over 65. I'm with you guys. I'm on the high risk category, and I give a COVID actually class this quarter, and there are papers out now that show that your brain, if you have COVID, shrinks. You have a smaller brain volume and smaller brain thickness. It's proven, it's significant. It's not huge, but you can detect it. Okay. Also, there's massive cardiovascular problems that occur. And so, regardless if there's another variant, which there will be, don't even think about that. Just what we currently have with the millions of people that have had COVID, they call long haul COVID. I can tell you the true burden of this pandemic has not been revealed yet. Okay, the people that are going to have to be treated for this is going to be, and we don't have anything clue about what's happening to our young population. Okay, but I can tell you with people over 50, your brains are smaller, and your cardio, it can be, every person, of course, but there is a significant difference in brain volume and thickness, and also a number of cardiovascular issues you've had. So I don't want to get it. I don't have to. If you got it, it's fine. It is what it is. I just wouldn't, don't fall for the trap. I got it and I'm okay. That's just not true. I mean, there's a couple papers out that show that if you've been boosted and have had COVID, you have the highest amount of antibodies. Well, all that study was done before Omicron. So it's not relevant. so until the study's done with the relevant bacterium or virus, I think we should still be vigilant. That's my opinion. So anyway, the bottom line from this is that we have 100% concordance with the fancy PCR machine that we did. Our answers in 25 minutes. Okay, it does not take hours, not take days, does not take somebody. If you can turn on your phone, you can do this. That's the technology that you need, and I can do that. I got mine right here. Okay, I can turn it on and off. Okay, the last thing I'm going to tell you about this test is that about its room temperature specimen storage. So it took us a while, but part of this magic mix that we made, we thought, God, if we could do it at room temperature, if we could now, and if we could store our samples at room temperature, this would really help resource limited nations, also at home too. Okay. And what this graph shows, or this figure shows, is that let's start with the room temperature. There's the blue circles. So we lose basically with our magic mix, we lose no, it's very, very stable to up to at least 4 hours. Then we lose after a day, 24 hours, it now drops. And if you refrigerate it, you get a tenfold drop up to a week. So you could actually, if you just have a fridge, you could just put your sample. And the bottom line is, you're testing massive populations, you might have to do that, right? And so this was, I think, going to be very important for it's called point of care testing, and also home-based testing. Okay, so I'm gonna get summarize what we've done. As I told you a little bit about this smart lamp, which is smartphone-based loop-mediated isothermal amplification. Told you that we've solved the primer-dimer problem, i.e. the false positive problem with lamp. Now lamp technology can be used in any setting, not just in resource-limited settings, not just with the smartphone, but with any. You can use PCR machines. You can do whatever machine you have. This will work. All it has to do is it has to have a single temperature that it can maintain. I mean, and it doesn't have to really. It doesn't have to be so cute either. Ours is from Walmart. I mean, it's not versus a $60,000 PCR machine. We told you that it's very highly sensitive and it's highly specific. We have 100% concordance with the clinical evaluation of PCR testing by the Cottage Hospital. I've just told you about the room temperature storage and component assembly. And I'm going to leave you some applications for home-based testing and point of care testing in resource limited nations. Also, what we've shown with this flu is that, we can rapidly adapt or die basically. And so this mix, and the question that we had earlier was a really good one, but we spent a lot of time, a year and a half on to have one, we want to make one mix that's going to work for everything that we can test. And we succeeded. And all you need to do is change the primers. And that is the cheapest part of this other than water. Primers are super cheap. And so you just have to design new primers for it. We have a new variant. And so our next steps, what we're doing is that it's our initial prototype was only for Android, a specific Android phone, the Samsung S9. That was which you get the time. We got it for $200, I think, $200 off Amazon. We bought like 5 of them. And now we're the next stage is to be able to have this technology for all Android phones, for all Apple phones. I mean, I have an Apple phone. I mean, that would be, it would just open up to everybody, not a specific phone, but we chose the Android because it's widely used and it's super cheap. Okay. And last is this, again, our vision was resource limited nations. Like right now, Africa is getting hammered right now. It's not okay. I think I know the vaccination rate for for just the initial 2 doses. No, for 1 dose, 14.8%. They're getting destroyed right now. I mean, this Omicron is just, it's horrible. Okay, so that was our vision, but we've now not changed our vision, but we've expanded our vision. And I'll tell you what really did it was my brother's situation. That is, where everybody in his family, all 40 people at his wife's birthday party got COVID. Okay, and this is 2022. That just should not happen in the United States. And so we really expanded our vision to, there will be a home-based test. You'll go to here's our vision. You'll go to CVS and you'll buy a kit, and it'll have, okay, wouldn't it be great instead of taking your kids to the doctors for everything they get, for strep throat, for staph infections, et cetera, you could have a kit. And again, the reaction mix is the same. You just change the primers, and you would buy these things. And we think we can get it down. Right now, we're at $6.47 a test at full price, buying the smallest quantities possible. That is the most expensive. And we initially, and I think we can get it down to $2, less than $2 a test. You get this thing less than $2 a test, you're going to buy a whole bunch of these things, and we're going to, it's going to save a lot of time going to the doctors, getting people out of school, people out of work. I think the future's now. I mean, this COVID is really, we've been thinking about this for many, many years. But it's just like going to the moon. Once you go there, you can see what you can do. Okay, and I can tell you that we've done a UTI test in 2018 using a similar technology for $1 a test, $1. Okay, and I'll tell you what we did. We went over to Cottage Hospital. This is started our collaboration, and we went over there like in 2016, I think, and still got the notes. And I said, hey, we got this test, and they're kind of looking at me like, yeah, right, whatever. And I said, hey, okay, let's go mano a mano. Let's take 10 UTIs. You get them, we get them, and just see who gets it. So it was kind of not fair. I had my guys ready; they're in the parking lot waiting for me, and so it was noon. I left at noon, and our test at the time took 50 minutes, and usually an UTI takes 18 to 28 hours. Okay, if you go through Cottage Hospital, it's 18 to 28 hours. So I called them up at 1 o'clock, and I said, hey, how you guys doing? They just got back from lunch. He said, oh yeah, it's great. Samples are in the queue, whatever. I said, well, we're done. Okay, good for you. You have a nice lunch. And I'm going to send you a PDF file. Don't open it. Open it when you get your results. So 28 hours later, they got the 10, and we matched 10 out of 10. So that's how we had. So they were really very generous in their time and been helpful in many things. So that's how we hooked them. That was, but it was really fair. I got my guys in my parking lot. I drove in, and they just had everything ready, and we just hit it. And but anyway, it got us to. So when we asked about COVID, they said yes. So I want to give acknowledgement for this part of the talk. Doug Hightoft basically ran the show. Lucian Barnes is now got his PhD. My son, Scott Mahan, was actively involved in this. Our computer scientist was Gary Fox. We're biologists, we just grabbed a person from computer science and say, we had an idea of what we needed. I didn't know how to do it, but we just said, and they said, sure, let's do it. And then two professors that have been super great, David Lowe and Chuck Samuel, and Cottage Hospital, or without them, we don't get this done. Okay, they also help fund some of this to Lynn Fitzgibbons. She's the head of infectious diseases at Cottage Hospital. Jeff Reed's head of critical care, and then three residents, Kat Arn, Andrew Bishop, and Sarah Edinger. And so let's answer some chats. I do have another test regarding sepsis that I want to go through if there's time. If not, that's fine. So let's answer the chats and get that out of the way if that sounds good.
Great.
Anyway, thank you.
Do you want me to read the questions from the chat?
Yeah, go for it. Whatever you want to do. Yeah.
Okay. Let's see. Mary asked, 25 degrees is room temperature here, but not in tropical countries. How high can the storage temperature go?
We don't know. Very good question. You're absolutely right. We haven't done those experiments. Those are going to have to be done. And how high can you go? And how long will it last? We don't know the answer to that. That's a really good question. So on the books. Okay.
Tom said, please describe the primer determination in manufacturing.
That's a good question. So what you have to do a little bit of, you have to be a biologist, you have to hire a biologist. The most important thing is you want to keep away from targets, gene targets that you think will mutate. We knew the spike was going to mutate. Some very big companies, it was just, really, it was, everyone's in a race. You do things you would never do. I get all that. And we did the same. But I'll tell you what we spent a long time on trying to figure it, to just, you don't know till you look through the literature, you see how things are mutating, and you can look at the genome, and there's programs for that. And we picked targets that don't mutate as much as the other. So you have to have a little biology to understand to do that, but basically just research it. And the primer manufacturing, I will never make a primer. They are so cheap, and they come the next day in the mail. You just tell them they're just, all they are is a small, let's say 17 nucleotides in a row, and they just lay on the RNA and they start the amplification, and they're super cheap. So I would never, there's companies that manufacture it. I would never do it myself. Or of course, if you're making a home-based test, et cetera, then you have to think about who to purchase from, et cetera. I get that. So that's the answer to that one.
Mary asked, so we can presume a primer can also be developed for noroviruses, or maybe it's already available?
No, it's not available, but that would be one of our next targets, would be that for sure. That's one of the biggest virus burden in the world. It's very common on cruise ships, for instance. And it'd be great to be able to have something that could detect norovirus. We chose SARS-CoV-2 for obvious reasons. There's a pandemic, and influenza, which is I would say, the people talk about for sure bioterrorism, that's the top on their list. And so we chose those two to start with. But yes, that would be great. So, we imagine the future though. You'll go to CVS and it'll just be a packet, and you just pick what you want, right? You just buy, so that will happen. This is not the Flintstones. It's over now. We have technology that we can do at home. It was not available before January 28th, 2022. It is now available, and all the stuff's out there freely available for anybody to work on, including ourselves. So
so RSV and other things like that.
Yes, no, RSV is very important. That actually probably be our next one. If you're going to go on the line, that would be the next one that we would do.
Mm-hmm. Let's see, Wynette, did you have a question here? Wynette says, huge implications for caregivers in so many situations, pediatrics to geriatrics to hospice, for everyone to be safe and able to eliminate some of the gowning, masking, visiting restrictions.
That's just a super great point. You know what, I'll tell you, and you guys, as you guys know, I mean, I'm at these care facilities, it's such a huge thing to take these people to the hospital. Okay, you know, it's like, we get it, right? I mean, it really, so one of our early targets will be at healthcare facilities outside of the hospital, because these people are ambulatory, right? I mean, like, to be able to get these, and the other option is spit in a cup and do the PCR, and it comes back a couple of days later, and you know, the whole freaking wing is infected. You know what I mean? So
Well,
if you could do it right,
risk our health with trying to help them, it was heart-wrenching. I'm an end-of-life hospice doula, and you know, the gamble that you take to go help someone and their family, they can't see them. So yeah, thank you.
no, I'm glad that would be one of our number one targets would be for that. And for that reason, my brother, my youngest brother, same one that got COVID, he's actually just became a deacon, and he goes to these facilities and, you know, provides a service, and actually just basically listens to some people, no one ever comes to visit them, you know, whatever. And he said, there's just no way some of these people are going to take it to the hospital. And so anyway, so I get, you're definitely preaching to the choir on that one. So,
Okay, let's see. Robert Muller says, could your test be better with false positives than PCR? Didn't Kerry Mullis say he wouldn't use PCR for public health testing? I don't.
how could you be? Well, I'll tell you, PCR regarding accuracy meaning do you have it or not is the gold standard. Now, okay, now I don't know the list why Kerry Mullis, who invented PCR, why he wouldn't use it for public health testing. I can tell you one thing, it's freaking expensive. I mean, PCR will never, ever be a home test. It is too expensive. You cannot get it down lower. If it would, it'd already be there. PCR's been around forever, since I was a freaking grad student. I mean, I'm an old guy. You know, it's like, it's not going to happen. It's never going to happen. And I'll tell you, at some point, someone's going to have to pay the bill for this. These free PCR tests, they ain't free. I mean, you know, that bill's coming. Let me tell you, maybe we won't have to pay. Maybe our kids are going to pay for it, their grandkids, or someone's going to pay for this. And but I think that we're at a place now where that we have other technologies that are as sensitive and as accurate, you know, and we don't have to, you know, buy these. Not to, you know, I started my own company. I'm not an anti-company person. That's not me, and I'm not conspiracy theorist, whatever. I'm not that guy. But I can tell you, this has been a pretty big money grab. I mean, you know, why would you use this new lamp if you can spend 20 times as much, get 20? So I mean, there's a huge incentive not to do it as well. And that's one of the reasons, to be frank, why we did it open source. Number one reason was we just want to do something good for the pandemic. That's number one. Okay. Number two was, you know, I was the CEO of our company, then I was the president, then I was dumped because I don't have the skills, got dumped down to the whatever board of directors or whatever. And we met with the largest, we were trying to get purchased. This is right after 9/11. Actually, that 9/11 killed us, and we were hemorrhaging to death. In fact, that year, there wasn't one IPO in the United States. That has never happened in history. For 1 year, there was not one IPO, and we were trying to get, you know, we were trying to get purchased. But we met with the largest companies in the world, you know, Pfizer, Eli Lilly, you name your, you know, we're trying to get purchased. That was because we were going down. And they said, hey, you know, we were with, well, I can't tell you one, but with one of the major companies that you would all know. It was between us and somebody else. And we had our CEO at the time had an idea that the number was much higher than what they were willing to pay. And then he goes, well, let me tell you what a pharmaceutical company will do. They'll just slap a lawsuit on you and you'll just hemorrhage to death. This is at dinner. I went, oh my God. That kind of wrecked the dinner. But anyway, so that kind of stuck in my mind. That happened in like 2003, I think. That's when we went down, we went under. But also I could see that this could be an issue here, that there's so much invested in the PCR technology. Why would they do this? You know, and so that's why we gave it out. So you're gonna have to deal with it now. So it's not stopping. So anyway, long answer to your question. So
see, Robert asked, can you explain more details of how this lamp works? It seems the magic is in the chemistry. Can this be done by eye and dispense with the phone? What are the wavelengths that?
No, very super good questions. These are great questions. They do it by eye. In fact, most of the reactions are done by eye. It's just, you're just more sensitive if you do it by turn to the optics in a smartphone. They're great. In fact, it's called, they don't have to get technical, but it's called the dynamic range of the range of signal that you can detect. It's freaking better on your phone than my $45,000 PCR machine. Okay, and the reason for it is that how do you get somebody to pay $1,000 for a phone? People want to take pictures and they want them good. They want that Christmas picture looking good. Okay, you pay $1,000 for that. So this is one of the major drivers, in addition to internet speed and all that. I get it, but for phone technology is picture taking ability. So the phone is much better than your eye, and we used a Samsung S7 and a Samsung S9. Our Samsung S7s were $100 off Amazon, and then the S9s are like, they were 200, I think. Whatever, now they're 150, whatever they are now. But I tell you, that lamp originated from, because it's fast and it's ultra sensitive. It originated actually just by visual, by no smartphone, no detective device, your eye. And so it actually is used, has a long history of being used by the eye. The issue again is the false positive. That was the thing that no one could solve. And so now that's also open as well.
Sorry, the first half of my question, maybe I wasn't clear. Can you say more details? Somehow we're missing the details of how this works. How does this amplification happen?
Okay, it's very similar to PCR. PCR requires 2 targets. So sorry, 2 primers per target. And so as you start from, basically what it means, you start from one end of the DNA on one side, you start from the other end of the DNA on the other side, and then you meet together, and that's what PCR does. And it amplifies in between. That's PCR. LAMP is different. It's very similar, but the details are different. It requires 6 primers per target. So our COVID test, we have 2 targets. And why you want 2 targets? And some of the biggest companies in the world did not think about that and it caused a big problems. That if your target mutates, those primers don't work. So you have all these false negatives. And actually, it happened to Roche. They had the most expensive test. They have their machine is $100,000 to purchase for COVID, $100,000, and it takes somebody, really sophisticated to run it. This is okay, but one thing they could do large populations. They're the most scalable test in the world. Well, they had 1 target, and they got burnt. Okay, bad. And then the CDC changed its recommendations where you have to have at least 2 targets, and so that means that we have 12 primers. Okay, you can see that with your 2 primers. Let's just do apples to apples. 2 primers per target for PCR, 6 primers per target for LAMP. And because of the 6 primers, it's much more sensitive. But it increases the possibility that there's homologies between the 6 primers between themselves. It's called self-amplification. That will amplify and does amplify if there's no other target around, and you get false positives. So it's very similar conceptually to PCR, but with 6 primers per target versus 2. Is that okay? Is that detailed enough?
Well, I guess, so why does PCR take so much longer than the LAMP amplification?
Oh, because yeah, great question. Because you have these 6 primers, it enables you to basically start these independent exponential reactions that now basically lead to lower time. I mean, it's in fact, if you just, I could be honest with you, it's pretty complicated actually. If you can get many YouTube, you just look up LAMP, hit Google LAMP amplification, and then you'll get like 100 of them, and you can go through and look at so the details wise, it is actually. But that's all the details. It's not like you have to do anything. It's just a matter of, but it is 10 to 100 times more sensitive than PCR because of the way it amplifies. But it's conceptually very similar, actually.
Okay.
Okay?
I had a question, and that was the CDC has a wastewater surveillance.
Yep.
They have a map. The closest dot to us is Southern Ventura.
Yep.
And I was wondering if this is, your test is something that maybe you've considered approaching one of the sewage treatment plants.
No,
or
absolutely.
something?
That is, all these types of applications are possible, and it's going to really reduce the costs of and the speed, and also speed is cost, as we all know. What we're really focused on, what my lab is focused on, is that we think that to be able to, we're doing two things that we think that are important, is that we're going to try to make this for any phone. So you don't have to worry about what phone you have. It's going to recognize the phone you have and just do it. Okay. And that's number one. Number two is that we don't have, it's called an EUA, Emergency Use Authorization. However, this paper has it met and exceeded all FDA requirements to get a EUA. We just didn't, it costs money, you have to hire attorneys and, blah, blah, blah. And so we haven't done it, but we think that's going to be important. So those are our two top priorities. And then we're going to now, and now also the issue, what we're going to be focused on is home-based testing. I think that it's silly, actually, with the technology that we have now, to not be able to have this available to everybody for, this $100 to $150 a test. It's absurd. And also think about it, guys, that's for one test. Okay. If it's $2 a test, you can take it 3 times a day. And also you just spit into a cup. You're not jamming a, rod up your telephone pole up your nose every day. It's just, we have the technology to do it now. So we're really focused on that aspect, actually. So it has a lot of applications, but that's where we're going to focus our efforts. Again, it's all open, open source. I mean, do what they want. That's we're gonna be focused on, and we're trying to find, funding to do this. Heck, there was no funding for this. College Cottage was very generous and gave us some money for the samples, et cetera, but it wasn't the cost of doing this. And I was just, what I did was hemorrhaging off other grants, and we need funding to be able to do all these things. And so, that's where, it's just the way it is. Like to see this.
Other questions? I think a lot of the things are kind of statements and
Okay.
saying how important this is.
Okay.
I'm just opening it to people who want to unmute themselves and ask questions.
Well, if there is no more
Okay.
questions, I'd like to tell you quickly about another test that's just have 2 slides that just came out this month for sepsis. If you want it, if you don't want it, that's fine.
No,
Is that
go ahead. Yeah.
Okay. So I thought that your audience might want to hear about this. Basically, sepsis is the #1 cause of death in U.S. hospitals. There's no number 2. It's sepsis. Okay. It's the most common cause of death in U.S. hospitals, 25% mortality. Okay, however, I don't understand this, but people think, oh yeah, I had sepsis, I'm fine. You're not fine. Okay, you have this leads to lifelong debilitation and cognitive decline. The reason for that is that you have clots. You can have clots throughout your body. That caused irreparable damage. Okay, and your lifespan's been shortened. Okay, so again, we were in the most advanced medical country, capability in the world, and 25% of our patients died of severe sepsis, and we still have these huge issues for the #1 cause of death in this country. It's sad. Okay, so what causes sepsis? Sepsis is caused by friendly fire. It's the body's overreaction to infection. So when you get infected, it's not the bug that's killing you, it's your response to the bug that's killing you. Okay, and what's happening, you get this friendly fire leads to severe inflammation, blood clotting, and organ failure. Okay, that's sepsis. Okay, and there's 3 things that are important for sepsis to make you to be okay: time to treatment, time to treatment, and time to treatment. All the fancy equipment don't mean nothing. The earlier you get on antibiotics is by far the most important thing you can do. In fact, when you're in sepsis, every hour that you delay treatment, antibiotic treatment, you have a 75% increased risk of death. Okay. So The current technology for sepsis is for diagnosis treatment. If you go to the best medical centers in the world, it's including Cottage Hospital, it's called a SOFA score. That's what the physicians are going to give you. They're going to assess you for a SOFA score. And what that stands for is sequential organ failure assessment. Okay, think about that. They're going to diagnose you for sepsis after your organs have failed. Okay, and so you're now treating after you have organ damage. Okay, so the answer is very simple. It's just been really hard. Is the need for early sepsis diagnosis and treatment before this injury occurs. All right, so this is 2022. This is the state of the art. Any physician in the audience, this is what they'll know. This is what they give, and they all know this. It's not like they don't know. They just don't have anything else. And I can tell you, so I don't know what the exact in 2022, but I can tell you our company, that Remedyne, that was in Santa Barbara in 1999 to 2003. Again, we were going down after 9/11, and we're trying to get purchased, and we met the largest companies in the world, pharmaceutical companies in the world, and they said, everyone said the same thing. The holy grail of biotech is to solve sepsis. More biotech companies have failed trying to solve sepsis than all human medical conditions combined. Okay, so look at, great, that was in 2003. We're at 2022. Nothing's changed. Okay, so I'm going to tell you that that we've now found some early markers, some major changes that occur in the blood right after you get infected, way before you have any kind of symptoms or damage. And it turns out a lot of them are going to be, we're going to show you data that certain coagulation factors rise very early after you get infected. They're not active yet. And then we can now use this as a predictor of sepsis. So once you have this event occurs where these coagulation factors rise, that physician will now know you will be in sepsis. I'm going to treat you now before you have organ damage. And we've done that. We've now led to early detection. This has all been done in mice. Early detection and treatment. This just came out. In fact, this is ahead of the issue. The issue hasn't even out yet, but that just came out this month online, and the issue's not even out yet. But we have early detection and treatment, and it improves mouse survival. So it's got 2 slides. What we did was really nothing in my lab is difficult. It's all simple. It's like, okay, I'm a geneticist. I'm also a little bit of an immunologist. We make vaccines and stuff like that. So I got to think that way. And what never made sense to me, okay, the sequential organ failure assessment, I get it. It's the only thing there. And we can't find any markers, but we thought, you know what? We're not smart enough to figure out, to predict what markers would arise really early that we could use as a test. Let's let the mouse tell us. And all we did, it was super simple. We just said, let's infect a mouse and see if there's any proteins, if there's any differential proteins in the blood that come up, that change well before there's disease. Pretty simple. So all we did was take a small aliquot of blood from the mouse, infected them with salmonella. Every day we looked at their blood, and look what happened. I got to give you a little bit about the infection process. You orally infect at day 0. You start showing a little symptoms at day 5, and day 8 to 10, you die. Okay. So this is when you've first seen symptoms is 5, and here is day 8. What we found is between day 3 and day 4, we had this catastrophic change in your blood proteins. Okay, look at this as you go across the board here. That's the massive changes. This is well before you have any symptoms or any damage to the body. So we said, so we kind of work with blood clotting. That's kind of one of our expertises. We're funded by the government, NIH to study. And what we found, we now look through, again, the previous slide of all those proteins. It was like 120 that we isolated. And we said, you know what? Let's look at the coagulation factors. And here they are right here, this part here. And let's start from the left-hand side. Again, you see between day 3 and day 4, all these major inflammatory proteins. There's massive changes. There's a lot to choose from, okay? So we chose coagulation factors because we know a lot about them. And again, day 5 is when you see symptoms. Day 8, when they start dying, day 8 to day 10. And so what we found is that all these are different coagulation factors, not just any coagulation factors, actually specific coagulation factors. And we now use that as these things arise in abundance, but they're not active. They're not active till well beyond here. So you have a lot of time once you've detected them to treat. And when you treat, they don't become active. And so we show a massive increase in survival in our mice. And what's exciting is that a lot of these coagulation factors that we've identified from previous, we now looked in the literature, and they also get increased in humans. So our next step is now to try this in humans. And so again, our quick summary on this is that, we think we have a way now for early sepsis diagnosis and treatment. There's major blood protein changes that occur right after infection. We know that coagulation factor abundance predicts sepsis. I just didn't show you the data. And we've now treated, because we now treat the mice as soon as we saw this occurred, the mice lived, okay? And they have way less organ damage. See, well, what about humans? What are we gonna do with this? Well, we're not saying we do this fancy blood protein test in every emergency room in the country. That's not what we're saying. What we're saying is that we're gonna now make a biopanel from those 120 or 200 proteins that change. We're gonna pick which ones are gonna be the best fit, and we're gonna put them into existing blood tests. So you go to the doctor, not because you're almost dead because you have sepsis, you go to the doctor because you don't feel right, okay? And so we think we can now incorporate these into existing blood tests. And we did this with at UCSB with Doug Hightoff runs my lab, my son Scott, who's now at Davis getting his PhD, another postdoc, Wan Ho Lang. So we did it with the Sanford Burnham Institute. And again, this came out just a couple of weeks ago. And also UCSD and the University of Sydney. But anyway, that's all I have for you. So if there's anybody wants any questions on that, or we're willing to entertain them.
Yeah, we'll open it up to questions. Go ahead.
Okay, how does that differ from cytokine storm effect, which seems to be a problem also with the COVID?
You're absolutely right. In fact, a lot of the markers that came up are cytokine related. These are this is called toll-like receptor signaling. This is the cytokine storm signaling mechanism right here. Okay, and you can see that there's a massive increase in this. And so yes, we detected that. The problem with it is that people have tried actually for the last 20 years to be able to use anti-cytokine antibodies. The problem is once the cytokines, they work so quickly that it's like once you detect them, they're already worked, they already signaled, right? I mean, they're so powerful. What's different about coagulation proteins, they're not active. They're not active till later, so you have time. With cytokines, you have no time. When it happens, you're screwed. Okay, and people have tried for 20 years, 30 years. It just can't, it hasn't been able to be done. Okay, but you're right, there's a massive cytokine storm, and we detect it. We have it right here. And I can go through if you like, the different cytokines, but the bottom line is that that's an important okay, I'll give you the real answer. You guys are smart. The real answer is why cytokines aren't being used is that, and it's been the biggest problem, is that the cytokine response is heterogeneous in the human population. So one type of the sepsis syndrome is also heterogeneous. And it turns out that what'll happen in you may not happen in someone else. That's the problem. And so what we know is now what about the coagulation? That we maybe get hit with that as well. All we know is that what we've done and looking in the literature, that with the ones that many of the ones, I can tell you that actually 14 out of the top 25 markers that arise in human sepsis, we found them in our test. And so the coagulation function, what it says in a way, the coagulation proteins, the coagulation cascade is more similar, and it's maybe not as heterogeneous as the cytokine storm. Does that make sense? So but at the end of the day, so you're absolutely right. You made it. I'm telling you, your question is right on. Okay. At the end, when you're dying of sepsis, it is homogeneous. Okay. But it's heterogeneous early. And so what do you look for earlier? You look for late, you're going to be dead tomorrow. Right. I mean, and so that's been the real problem with using that as a signal. Does that make sense? I hope. Does that make sense?
Makes sense, yeah.
Okay, good. I thought I was not answering your question.
Okay, other questions?
The question I had is, are you suggesting that physicians don't already treat for infection very early when they even suspect sepsis? They don't wait for several days to get absolute proof, do they?
No, you're absolutely right. So I can tell you 50% of our samples, 50%, we've been working with them for a number of years now. We're part of this NIH consortium for sepsis. There's no bug ever identified. So you got to be smart. The physicians got to be smart. Now they'll treat as soon as they feel that with their experience. But again, it's their experience with no data, right? I mean, so would it be nice to have data to go along with that? Right? Yes. So the 2016, actually, there's this huge, massive study was when they changed the way that where it was determined that time to treatment was the most important thing. There was no number two. It's time, when did the patient get antibiotics? And that changed since in 2016. It changed the way physicians worked, because there's a lot of pressure on physicians not to reduce the antibiotic use, and also this lower doses, smaller durations. There's a huge pressure on them. There's actually a committee, like any major hospital, like Cottage Hospital, there's a committee that monitors all this stuff because they're trying to stop antibiotic resistance and stuff. You know, for good reason. I'm not saying it's called antibiotic stewardship. And what we showed here, actually, I didn't show you the picture because it's kind of complicated, but we showed that if you start antibiotics late, you can save the mouse, like you save the patient. But, which surprised me, we went early to late, and late we could save these mice. However, once you stopped the treatment, the disease relapsed. So what it says is that there's blood clotting, there's physical changes in that mouse, that those antibiotics don't work anymore. You can't clear, right? So we said in our paper, and actually we got to, they self-identified who they were. They self-identified themselves as intensivists, i.e. they are MDs that work in a hospital in the critical care division. Those are intensivists. They had the same, he goes, are you saying that we need to have a longer duration? I said, that's exactly what we're saying. And he goes, you got to put that in the abstract. You got to put that, I mean, we didn't have it. They thought that was a major point, but anyway, so yes.
Thank you.
Is that, so physicians do it as early as they can, but wouldn't it be nice to know, I'm talking about way before a physician physician only does it if you have all these major symptoms. What if you could do it before that happened? That's what I'm saying, right? We're talking about a couple of days before, right? I mean, but when a physician has enough evidence to treat you, you have damage, right? They're not going to give you antibiotics because you go in because whatever, you have a fever or whatever. They're not going to do that because you might have the flu, right? The antibiotics aren't going to do anything, and you might react. You get no benefits and get all the risks of taking antibiotics, right? I mean, so there's huge challenges for physicians. Is that
They're
fair?
having to weigh the comorbidities, the side effects, everything else as all of this is unraveling, right? With the patient.
Absolutely, yeah.
Yeah.
Okay.
Oh, yeah. Oh, sorry. We have 3 chats.
Yeah, you can read those. I think I'm not seeing too many other questions in the chat. So
Okay.
if anybody has a question, you can ask it now, or otherwise we'll just thank you very much for your talk. It was very informative. So.
Well, I'm good. Well, thank you very much for having me. And it was really, I thought the questions were awesome. And And it was just simply my pleasure. So
Yeah, I'm looking forward to the availability of this test. Robert, did you? Oh, that was clapping, not a raising hand. Okay.
anyway, have a great day. And thank you for everything. That was great. That was fun.
Okay, thank
All
you.
right. See you.
Thank you.
Bye, everyone.